We are recording now. Welcome to the evening of Python coding. And today we have a very special presentation by Patricia Mason. She's going to show us a library that relates to Python and biology. Python is emerging as one of the languages that deals with biology data more than other languages that I'm familiar with. I see it quite a lot these days. So today we're going to show one library that's related to one biological database. It's called CHEMBL, Warehouse Client. I think it's pronounced ChEMBL, but I'm not sure how it's pronounced. But without much ado, maybe Patricia will tell us exactly how to pronounce it or how she thinks it should be pronounced.
You can see the first slide, okay?
Yeah, we can see the slide. We just don't see you. Well, if you could, wait a second, I'll make sure that's like, I'm going to pin you for a second so we can see. Here we go. Now we can see you, and now I'm going to unpin you and go back to the presentation.
What's the verdict on how to pronounce it?
You know, that's a toss-up. I haven't really met anybody from there. I have only ever dealt with the EMBL back in the day, and I'm assuming that this is a CHEMBL web resource client, but you know, I probably offended a lot of people just by saying that. My name is Patricia Mason. This is my email address if you'd like to contact me at any time. I have a BS in biology from Santa Clara University in California. I started off working at Stanford University in the microbiology and immunology department. At the time, we were doing some of the first generation DNA sequencing. So I was really involved in genomics in that lab. Specifically, the lab was looking at the major histocompatibility complex of the immune system. So pretty much all of the genes that are in the immune system. And we were also going trans-species. So interestingly, I was working on the trout genome, and I did that for about 3 years. And then I joined what was considered one of the first bioinformatics companies. And that was a new word at the time. And I still think it's a very vague word. It can mean a lot of things. But at the time, it meant analyzing genome sequences. And this company at the time wanted to document and patent every genomic sequence that they could find, and they did do that. And if you work with patents, you know that those only last 7 years. But their thought was, if we were doing gene therapy, that they would end up making a lot of money. They ended up closing and getting bought out by another pharmaceutical company because the thought of gene therapy kind of went by the wayside for a few years. After that, I went into STEM education. I taught kids about math and science. And about 2 years ago, I got together with a friend. Both of us wanted to get back into this field of bioinformatics. And we had both decided to take a class through Coursera, which is a really nice online education platform, and the class was through UC Davis, and we were to learn bioinformatics and DNA analysis alongside of learning Python. Well, both of us, along with a lot of other people that I read in messages, realized very quickly that we had to learn Python before we could go on with this class. So I started learning Python in earnest. It took me about a year to really catch up, find a good platform for myself to learn on. I can recommend platforms, but I think everybody learns differently, and if it's your first language, you probably learn differently than if it's your second or third language. But I did spend quite a bit of time learning Python, and last year I joined this group, and someone had given a talk sort of in the data science realm, and we got into discussion about data science and how Python can be used for that. And also at the same time, Austin is not really known for its huge amount of bioinformatics or biotech companies. So I decided to broaden my scope a little bit. I did a data science boot camp with General Assembly, that's the name of my boot camp. And I graduated in December with that. I did focus my capstone in bioinformatics a little bit. And what I'm showing you was part of the capstone that I wanted to do, but I never really got to. You know, these capstones can just go on and on and on, and I didn't have that much time, but I really did enjoy this part of it. So what is bioinformatics? And like I said, that's a really loaded question. If you look at Wikipedia, it says bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data, which is a mouthful to begin with. There's so many things you could do. But the next part of the sentence is, in particular, when data sets are large and complex, and data sets have gotten massive. When I was initially working with genomics, they hadn't even completed the Human Genome Project, and much less than any other species. And now they have a lot of standard species fully sequenced, and I mean, you can almost, I think you can order your own DNA sequence now. It's a lot more inexpensive, and just like with computers, what we would work with these well plates to sequence are now on small little microchips. So there's just massive amounts of data, and then you add on layers. This next sentence is describing, it's an interdisciplinary field of science. It combines biology, computer science, information engineering, mathematics, statistics, and then a lot of biotech now is synthesizing molecules or synthesizing DNA strands. And what you're seeing right here on this part of the screen is an alignment of protein sequences, and this is probably very similar to what I was doing both at Stanford and inside genomics. What I would be looking at actually the DNA. This is the protein, but we would align sequences, compare the parts that are the same, compare the parts that are different. Sometimes there are proteins or nucleotides that are inserted that cause issues, and that's when you get into diagnostics. There's a company here, they're both in San Francisco Bay Area and in Austin, they're called Invitae. They do a lot of genomic diagnostics where if someone has a disease, they can take it all the way down to their genomic DNA and find out, you know, do they have a mutation, an insertion or deletion that's causing their particular disease. The part of the talk, it comes from the European Molecular Biology Association, and in a branch of it is called the European Bioinformatics Institute. The EMBL has been around for a long time, but they've expanded greatly. They've collected a lot of data, as they say here in this slide, that helps scientists realize the potential of big data in biology. There's just so many things that can be done, and there's so many parts of bioinformatics now. They have branched out into chemoinformatics, proteomics, as well as genomics. So there's all these what people call omics in this industry, and that has created enormous amounts of data. But this is one of the places that houses this data. In particular, they house what's called the ChEMBL or the CHPMDL database. It's a manually curated database of bioactive molecules with drug-like properties. This brings together chemical, bioactivity, and genomic data so that they can use genomic data and bioactivity together, which is a relatively new thing. It used to be a completely separate entity, but now because of all of these different machine learning and data science activities, they can actually analyze bioactivity and tie it to genomic data. And this web resource client, it says this is the only official Python client library developed and supported by the ChEMBL group. It says you don't need to know how to write SQL, which is great. It says you don't need to know how to interact with the REST APIs, even better. You don't need to compile or install any Bioinformatics frameworks, perfect. It handles interaction with your HTTPS protocol. And the best part of this, it says it also implements lazy evaluation of results, perfect for biologists. And you only need to evaluate a request for data when the value is required. So this it checks all the boxes. And I'm showing you this slide because this is something that if we're going to a talk, a biologist is always going to throw something like this up, and it usually scares people, but it's just a cascade of activity from the outside of a cell to all the way down to the DNA. And the reason why I'm showing you this is there's this GRB2 molecule. It's one of the molecules that I was interested in for my capstone. I was studying ulcerative colitis and Crohn's disease, and this is part of the pathway that's activated when a Crohn's patient or an ulcerative colitis patient is having an episode. So let me go and show you how this GRB2 is. I can search for it using this CHEMDL database.
What's GLB2? It's a molecule. It's a gene. It's a what?
Protein. It's a protein. It activates other proteins. It's involved in what's called a cascade of proteins. So you have the ligand on the outside is bound to a receptor, and that receptor, the P stands for phosphorylation. So you have, just to make it simple, you have this cascade of activity. So it's almost like passing the baton at the Olympics. All of these molecules are passing the baton to the next molecule until you get all the way down to the nucleus, and that turns on gene transcription in some way. It can also turn off gene transcription. It really depends on the pathway.
And transcription means for non-biologists like me?
It means making more. It means making a protein essentially.
It's like duplication of the gene, or it's like the activity that the gene makes proteins. This is what you're saying?
Yes, it's essentially the gene making the protein that it needs at that time.
Okay, so this GLB2 activates the fact that the gene will eventually produce another protein, if I understood correctly.
Yes, the GRB2 activates something else, that activates something else, that activates something else, that goes all the way down into the nucleus and causes transcription and down the line a protein.
Okay.
Sometimes in cancer, this happens and you get a protein that you do not want. So you may want to inhibit this activity. Sometimes you want to activate this activity if your body is not.
So just to give us an idea, how many of those activities or proteins exist in the human body? Like, just give me numbers, like thousands, hundreds of thousands, millions?
Okay. The human genome consists of about 30,000 genes. I don't know how many genes are expressed in an individual.
I'm trying.
I know that.
I'm asking this for a reason, just to explain how big the problem is if you try to program things, how big of databases and stuff like this you need to have and stuff like that. So It's millions, it's tens of thousands.
Well, genes are never straightforward either. You have different, you can put together a gene in different ways. Sometimes genes, there's called isoforms. So even though you have a gene that makes a protein, you can have that protein look a little bit differently. And that's what makes this difficult, is that, yet we do have 30,000 genes, but each one of those genes can actually be 5 different proteins. And definitely we don't express all those at the same time. For instance, in utero, in an embryo or a newly fertilized egg, it's probably the most amount of genes that you'll be expressing in your entire lifetime. Most of those get turned off by the time you're born, and even more into adulthood. You don't want to be expressing those genes.
So basically you're telling us there's like 100,000, hundreds of thousands of proteins that are of interest. And then modeling the interactions between them, maybe, well, hundreds of thousands times hundreds of thousands makes like much more types of interactions. Am I correct?
Well, you're talking, there's other molecules in your body besides proteins. There's, obviously carbohydrates, you have fats, and proteins are just part of that, even though they make up a large part of your cell. But we do have those other parts of our bodies, or other parts of the cell that exist. So yeah, you could be talking about a lot of different molecules, but in this cascade, all of these are proteins.
Okay, thank you.
Anybody else have any other questions before I go on?
Patricia had a question about having a database you need. I'm not sure if you got to that.
No, I have not gotten to the database. Okay. Can you see the Jupyter Notebook okay?
Yeah.
Okay.
And people can download it from the link I put in the meetup. So you can actually look at it on GitHub or download it, or she's now showing it from a local machine.
Yes. You can click on this link that I have, the GitHub of the ChEMBL resource client. I adapted this demo directly from their GitHub. You can pip install this ChEMBL web resource client. I don't know if you can Honda install. I usually just do what people tell me. So.
For people new to Python, explain what pip install means.
Installing this library.
So Python.
Is that basic?
So
I can get.
Yeah. So Python comes with a set of libraries, but it can to allow more functionality. It allows you to bring in more libraries from a source called PyPy, PyPI, and pip install just takes the library from there and puts it in your machine, in short. Sorry, go ahead.
That's okay, it's great. If anybody has anything to add, please let me know. I am certainly not an expert in this part. To use this library, you need pandas, so I imported pandas.
Explain what pandas is.
As far as I know, it's a data frame manipulation library. That's all I've ever used it for.
So basically, does what? It stores tables or something like this?
I think more table manipulation. You can access certain parts of the table with pandas. That's what I use it for.
Okay.
So you can take, I showed you this GRB2. You can take this new client, say that you want to target a gene, you can put that target into a variable, and then you can do a search like you can for this gene, and call it a query, and that returns a dictionary of information. You can take that dictionary and turn it into a data frame, and you can see what this data frame returns from this ChEMBL web resource client. And it looks like a bunch of gobbledygook, but it's very useful because it tells you what organism it comes from. It tells you what the actual preferred name is. It gives a score. I think it's just the matching score. It gives an ID from the ChEMBL database. And it gives like a target type and another ID. You can also do a
What's musculus, and why doesn't it return 3 records for the same gene?
I'm sorry, what was the second part of the question?
What's organism? It says musculus.
That's a mouse. That's a common mouse.
Good. And why does it return 3 records when you ask for 1 gene?
These are all the genes that are stored. Like I said before, that this gene exists trans-species, and so they will sequence this particular gene in different species. Just like I mentioned before, that I was looking for immunological genes in the trout. They do the same thing across all organisms, and I'm actually surprised that you don't see more. Usually, if you do a search, for instance, in the NIH, will come up with dog, cat, cow, usually mouse. There's a set of standard organisms that they will sequence genes in.
Okay.
And so this is not a really well-known gene. It was of interest to me just because it was in this cascade that I was looking at. And you will normally find, if there's something very specific that you are looking for, you will not find that it has been sequenced in a lot of different organisms, or that it's been identified. It might have been sequenced, but it hasn't been actually identified in a lot of other organisms, or it may not exist. But that is actually standard. You could also do a synonym, and the reason why I had this commented out is this takes forever to do, and I was debating whether to do a live demo, but it does take forever. You can do just a sort of a synonym or just a general search for names.
So it will look for the entire database of things that look like the GRB2 and bring you more records?
Yeah, it will expand the search, and you will get everything that says GRB2.
Okay.
If you want to get your selected target, say I wanted to look at this mouse, GRB2, you could pull out your selected target. And then, yes.
What's the selected target? It's like how to select a specific row, or?
Yes, if you want to select the first row, you could select the first row and get that target. Then, if you wanted to get, say, the bioactivity for GRB2 in the mouse, you could do a new client activity. You could pass that to a variable, and then you see where it says activity.filter. You could filter for different aspects of this database. And you can also stack filters. You can do .filter. You can access different parts of the database. But I only did one, so I did the selected target. And
so explain the activities. So the gene does its interactions with other genes or something like this, or?
No, okay, let me go down here. This is the database. So you can have, this is the information from the database. You have a lot of different things. I'm not a chemoinformatics expert. I really was interested in this, so I started to look into this. if everybody can see this canonical smiles right here, canonical smiles, and if you've ever seen one of those 3D structures of a molecule where it kind of looks like Legos or something like that, you know, like little circles that are connected by toothpicks, something like that. Canonical smiles are 2D structures. They're 3D structures smashed down so that the database can handle them. And they actually do solubility studies on them. Say if they're looking for a drug they want to make, and they want to know if a certain molecule is soluble, or to what extent it is soluble. There is another Python package that you can use further down the line on this. This is just sort of the beginning of what you could do. There's another package called RDKit, and it actually analyzes further down the line a lot of the things that you see in this database. Let me see, what else does it analyze? There's a few values in here. There's a lot of things in here that you can analyze with the RDKit, and I won't go into that because it's a very, very powerful package, and it's something that they use for drug discovery. Does anybody have any questions about this database?
Well, that's a lot to ask. It's like, it's huge, but
it's a bit overwhelming, and like I said, it's so much information, and that's the very interesting part of this. You know, you can almost make your own discoveries. This is a database that has hardly been tapped at all. They just put it out there for people to play with.
So just to explain, and I want to expand on the point that you're now just saying, this is so people will understand. This is a new frontier. And let's ask you, if you didn't have Python, this database, how much work you'll have to do in whatever you were doing research or in your work before, how much more work would you have to do if this didn't exist? Or how much this simplifies things for you?
I used to do a lot of my work, and everything was sort of on a web-based server. And I just think with the amount of data that there is, and you're limited to whoever created the web-based server, whatever they wanted, there was no interface to create your own queries or your own scripts or anything like that. This just makes it a little bit easier for that. And then also machine learning, a lot of people do modeling on this, so it's hard to interface all of that on a web server. Like if you were to go further on down the line with RDKit, you would have to use machine learning programs, statistical programs, such as sklearn, which I know a lot of you use. And to get those all in one place is a very difficult thing to do with databases that are this size.
So you're telling us basically that it opens new opportunities that were not possible before, and many of them are, the interface is Python. This is what you're telling us.
Yes, the interface is Python, and it just makes things more flexible to do a lot of different things, especially with chemoinformatics, just because genomics, I feel that they really have that part of the omics down. We've been aligning sequences forever. We've been sequencing DNA forever. But now that we're into the chemoinformatics side of biology, and it spans so many different aspects of machine learning and statistical analysis, a lot of that is very difficult to do. I mean, I'm just trying to think, like, if I were to put that in Tableau, if you would put it in anything else to do the statistical learning that you would need to do, it would be very difficult.
Yeah, I agree. this is why it's a new frontier. Really, the fact that you now have a database you can get the data from, and this is the ChEMBL is one of them, and you bring it into a language like Python that has very good tools to do machine learning. You can automate many things that in the past you have to put a person to actually do the things by hand. Think about this. Only you write programs that will do sweep things and do things automatically that humans used to do by hand. This is, am I correct, Patricia?
Yes, exactly. I mean, it would be a little bit, I mean, I used databases when I was at Stanford. I was in charge of the HLA database, and I did write very simple queries, but I could have done so much more if I had Python and a few more analysis tools. And just because so many people work with DNA sciences and Python, and just people are always developing tools, and they're so easy to install. BioPython, I'm thinking that's another one that's a very good tool. That's used mostly for genomic data, but this chemoinformatics field is so much different now. Like I mentioned before about the canonical smiles and trying to figure out solubility, and there's something called Lipinski factors, and just so many things that you can use to analyze different parts of the molecule that weren't readily available before, or that you would have to run analysis by hand, or you would have to do something in the lab by hand, that now you could do computationally, or you could predict something computationally that only before you could do in the lab.
So, and just to get you an idea, lab experiments take forever, and they are costly, materials and stuff like that, and you have to put a person on it. Think about the ability to bypass all this.
Well, let me just give you an idea. When I was working at Stanford, the graduate students that I was working with, and they would be there for 4 years, it would take them 4 years to determine the structure of a protein, and these people lived in the lab. Absolutely lived there. I mean, they did nothing but eat, sleep, and try to figure out the structure of their protein. And now, I don't even think it takes people nearly that long to do it. I haven't really talked to anybody, but it's a lot faster now. They can do a lot more predictions. It just makes things so much easier.
I have another question for you. What are long-term, like in the human words, something simple? What this can be used for? What are applications that are possible with all this new knowledge and all those Python tools? And give us just one or two examples. What can it be done? And I'm not talking about low-level things that you need in the biologist to understand. Something that simple people can understand.
Okay. If you see this assay description right here, these assays used to take probably a week in the lab. And I think the key here is something called high throughput. Now they're able to do these things simultaneously in small little wells, and they can study the bioactivity of many different compounds at the same time, and then put them into a database like this. Whereas before, it would take one person 1 week to analyze one compound. Does that make sense?
Okay, but what is it? My question is higher than the data that you see here. This data that we're here seeing, what can be used for? Can it be used to calculate things like, here now we have COVID, can it be used for vaccination? Can it be used to drug treatment? Can it be used to understand something that's happening in the body? I have no idea. Like, you're the biologist. What can be done with this data that wasn't available before?
Sure. As far as I know, it's all bioactivity. So it's everything that's currently happening in a cell. So it's real-time, actual processes that are happening in the cell that can be looked at, whereas before you would have to take slices of time in the cell. You would have to, for instance, lyse your cell. You would have to get the items out of your cell.
So
and
now
I hope I'm.
So now you can simulate it. This is what you're saying.
Yeah. Yes. Now
Okay.
you can simulate it.
And if you understand the cell better, understand the cell better, what can you do with this knowledge? What it can be used for applications for understanding the cell better?
Well, for instance, this GRB2, if you have Crohn's disease and we can assay 1,000 compounds in an hour and figure out which of those 1,000 compounds can prevent this GRB2 from, for better, lack of a better word, wreaking havoc in your body, that's much faster than one person, 1 week, analyzing one compound.
And this can lead to new treatments.
Yes, new treatment.
Good. So the availability of this data with Python will accelerate our ability to understand the phenomena and treat biology better and treat diseases and stuff like that. Correct?
Yes.
Good.
Yes.
Okay. This is what I was aiming at. Something like high level. But anyone has more questions other than me? Okay, everyone is a biologist here and understood everything apparently.
It looks like Sagar had a question. Can this be used to understand GMO crops?
GMO crops? Is that the question?
The question is, can this be used to understand GMO crops to predict how to grow, for example, seedless apples?
If the gene.
What's GMO crops for all of us? Because we don't understand what it means.
Are you talking about genetically modified organisms, correct?
Yep, that's what I was referring to. Thank you.
Yes, to genetically modified organisms. Well, that is more of a genomics question than a chemoinformatics question. They have been manipulating DNA for a very long time. And so I would say that, yes, on a high level, this is an omics question. However, that's probably more on the genomics side of the question.
Okay, gotcha. Thank you.
Sure.
Yeah, any more questions? Please don't be shy. And I know that I don't understand the half of it, and I've been working on fields that are close by. So please don't be shy. It's okay to ask questions.
Yeah, if there's you can ask anything.
Okay.
Okay. Well,
coronavirus. Yes, something.
Yes, everybody is fascinated with this, so I thought that I would search for coronavirus. So you could put in here a word if you'd like. Search for coronavirus, do the same thing, put it in a variable, get a dictionary of items. There are a lot more. You have feline coronavirus, you have human, they have different SARS that are associated with it. So you can get a few more things out of this search. And say, for instance, I decided to use the fifth one. Is that right? Oh no, sorry.
The fourth one.
Yeah.
Actually, it is the fifth one. It's just index number 4. It's the fifth one, but it's index 4.
Okay. So I did another target, looked at the activity, and there, as I told you before, if it's something that's well studied, you can get a lot of more information. The one that I was looking at doesn't look like too many people are interested in GRB2. Definitely a lot more people are interested in coronavirus at this time. So you're going to get a lot more entries that are in this, more assays that have been done. Trying to think of what else is in here. If you look at the type, so they do different types of studies. They do inhibition studies. They do what's called IC50. Don't ask me too much about that. I know that it has to do with solubility. Again, I'm not an expert. I don't know what some of these other studies are, but they are different types of studies that they're doing on coronavirus assays. So you can get more information from these.
Yeah, and there are many columns. It's not even, how many? 45 columns. Whoa.
Yeah, we're going to get the same thing as you got up here. So you're going to get all the same information. They even give you papers. They give you everything that you can possibly want.
So there is a question, I think two of those. So Manoj is asking, could you also find a diet that fits just based off genes of a person?
I'm sorry, what was the first part?
Could you also find out diets that can fit, basically, can you find the diet that fits a person using this, the genes, using such a database?
You know, I have a pretty strong opinion about that. I think it's hard unless you have an actual genetic component to the dietary problem. For instance, celiac disease, that's an actual genomic problem. That's an actual gene expression problem. That's an inherited issue. If some food sensitivities are also immunologically related, but a lot of that's environmental factors. So I think it is a little bit more difficult with dietary issues to nail that down. I suppose if you were to focus on that as a bioassay, it could be an avenue for determining, for instance, Crohn's disease is a gastrointestinal issue, and they have narrowed it down to T-cell activity. So I wouldn't say no all the way, but I think it would have to be a specific problem that you're trying to answer. Does that answer your question?
Well, I will have to go along with the question, with the answer, because we have another question. Brendan is asking, does that assay ChEMBL ID field let you find out when the data collection, assay in parentheses, was done? And is it different from the original sample? Is it different? And is that different from where the original sample was taken? I guess, broadly speaking, how does the data collection behind this data set work?
Well, as far as I know, it's like the NIH. People upload it to there. So usually these are from peer-reviewed papers. They have probably a little bit of quality assurance, but as you can see in here, if you want to go back and find out exactly about these assays, you can go to number 15, document journal, document year. You can get the documentation. And that's what I did when I was doing my capstone, is I went to a well-documented study of Crohn's disease, just to make sure that it was from a peer-reviewed paper, that the data had been seen before by other people. So yeah, there are ways to see where the data came from.
Okay. Patricia, you have more to show us, or?
I don't think, nope, I think that's it.
Okay, and are there any more questions? Please.
Hi, so I was listening in here, and I looked it up, and it looks like it was obtained from 34,000 publications and 12 chemistry journals. So it looks like it's a manually curated data set.
Yeah, they said that, yes.
They probably scan some things. I don't think they probably automated some of the work here.
Well,
But who knows?
I don't know if people have the access or the ability to upload their own data like they do with genomic data.
Yeah, because I was looking to see if they were grabbing it from Uniprot or anything else like that, but it looks like they were literally manually pulling it in and stuff. Because some of these other databases they use can get kind of large, and it's tough when it's all kind of put together in weird different ways.
Yeah.
Okay. Any more comments, questions? I have a question to you guys. Did you like the change towards biology? Less Python, more biology?
Very insightful.
Thank you.
Well, I do bioinformatics at work, so it was interesting to see another tool.
Really? Okay,
Yeah.
you'll be using this tool now?
I'm a computer scientist. I will be kicking it over to the bioinformatics people for them to take a look at and see if they use it. Because they spend a lot of time looking at different proteins and seeing, how it affects organisms and what exactly that protein does and how you can change that protein and how it affects it, and what it does to different people and different environments and stuff.
Nice. If it helps them, then let us know. We're
I
always
will.
glad to help. It's all volunteer based here, so it's like we like to have good feedback if there is some. Anything else? Anyone wants to add anything else? Questions going once? Questions or comments going twice?
Hey, I have a question.
Go ahead.
Well, it's just in general, when we're talking about this, are there any, ethics concerns with this?
Well, I will tell you this, any human metadata that is probably HIPAA restricted, any protected health information has been stripped. So, are you referring to that?
Yeah, it's just like the use of this stuff. I mean, I didn't know that till today. Yeah, just the use of this, I guess it could be targeted. Could you create applications to target certain people, whether that's for good or bad? I just wonder that type of thing. What sort of ethics concerns do we have with this biological data, or just basically the use of it?
Well, can you give me an example of
I think I know where he's getting at. Let me continue. So, there's a lot of those biology data available, and now the question is, can it be used for good or for bad? So, we want to use it for good. He's afraid that, are there any restrictions or people using it for bad? Because it's available. It's like, let's say if the chemical stuff that you buy in the store, then if it's poison, you have to say poison, or stuff like that. So it's like he's looking out the rules around that. If someone wants to use it or just explore this, then should they be aware of some ethics? So this is a very good question. The biology world will have to learn to adapt with it. Same with models, by the way. I do disease models. I have the same concerns. It's a very good question, James.
Yeah, there are ethics that are required. A lot of academic institutions follow a standard set of ethics. There are laws, like for instance, the WHO, they have a certain set of ethics also. That being said, I'm sure that there's things that are going on that people wouldn't agree with. So I think it just depends.
So if I, for example, tomorrow, something, I'm trying to think of something bad that's like, let's say here, someone was talking about the diet. Let's say I do something that is like, a little bit fishy, and I say I use this database to compute something that helps people lose weight and make money out of it. Is it ethical or not? What?
That's against some promotions where, I don't know, like you make a special type of popcorn or something that certain people, it makes people want to buy more popcorn. know, just like if you could this be used for, I understand that scientists don't necessarily understand this, but like somebody with a mind for science and business, they can say, well, I can use this to make money and sort of damn the ethics of how I do that. Like, is that possible?
There are a lot of, I mean, for instance, we have the FDA, a lot of countries have similar things. So, you know, doing research is one thing, selling it is another.
Good. So basically what you're saying is beware of what you're doing, don't do anything unethical, and eventually there are some regulators like the FDA that if you do something wrong, they will catch you.
Yeah, I mean, to get a drug to market, to get a food to market that you're making certain claims about, you know, there are certain hoops that you have to jump through.
So, this is good that now this information is available publicly. It can accelerate research into good things, but we also have to be aware of security issues or some other concerns of those who don't follow the correct path. But I believe that there will be oversight, because once those databases will be used for other things, I believe that the governments will know how to restrict things. I believe so, but hopefully they're already working on this and already have good plans for that. I know of some things that they're developing, but this is really new frontiers, and new frontiers are always problematic. But James has a very good question, and actually, James, your question relates also to computer science. Computer science itself as a whole is not really highly regulated. It's also an issue. But unless there are more questions, I suggest that we close the recording. So I'm gonna go questions first time, questions second time, questions third time. Okay, no questions? I don't see, wait. Yeah, someone said about a comment from LEFA, specimens collected without consent may not be used in such research. Yeah. And she also said that in research ethics require patients to give consent to specimens. Yes, I know, informed consent. And LEFA also said that HeLa cells lines are examples of potentially unethical use of patient specimens. Henrietta Lacks, do you understand what it means?
I think HeLa cells, that's a cell line, that's a very old cell line that they used. As far as I know, they're still using it, but it may have been collected illegally. I'm not surprised. Yeah.
What's a cell line? What's a cell line?
So you can take a cell and what's called immortalize it. You can infect it with a virus. I used to make these cell lines with Epstein-Barr virus, and it would make them propagate indefinitely as long as you put them in the right incubator with the right amount of food. You get this cell that will propagate itself forever.
Okay, so what's the Henrietta one?
I'm sorry, which one?
She said Henrietta.
It might be from
Henrietta Lacks was the patient that they got the cell line from.
Oh, from the HeLa. Okay, it might have been the person that they got the HeLa.
In the 1950s, and she had no idea that they had, created this immortal cell line, and then it's been a whole industry around, profiting from this.
But I mean, that's, which I find strange that they use her as an example. They take leftover pieces of people all the time from medical places and use them. So I'm surprised that they picked that one. I guess it's the most famous one. But
it is the most famous, and also I think it's for those who do ethical training, this case would be brought to our attention as potentially unethical.
Yeah.
Use.
Okay, this is Eric, correct?
Yes.
Okay. I hear.
I have to redo this ethics training every 2 years.
Actually, so Eric works with clinical trials. Okay, so he knows the details here. So thank you for the insight. Okay, I'm gonna go for the questions again. And by the way, after we finish the recording, please stay. We're not going away. So questions once or comments? Question or comments twice? Question or comments 3 times? Okay, we're stopping the recording. Oh, before we stop, Patricia, thank you. Please, you can unmute yourself, make some noise. It was so fascinating, I forgot to thank you. Well, this is very good. Thank you, Patricia. We're stopping the recording. Don't go away.